All right, in the next part of this chapter of learning what goes on inside the cell, we're going to take a look at protein synthesis, which this is an incredibly important part of the cell's job. So we're going to take a look at how protein is synthesized and, what DNA does and what RNA does and how that's related to protein synthesis. So if we look at DNA, we talked about DNA in Chapter 2. But remember, we've got two strands of nucleotides, So here, let me draw this here. There's one strand, there's a second strand. And remember, they're twisted and they're held together by those hydrogen bonds, That weak attraction, remember we talked about, that's holding it together and keeping it spiraled. The important thing here is we have 4 bases that are all paired together. Okay, so here's our 4 different bases: adenine, thymine, cytosine, and guanine, A, T, C, and G. And they pair together, in these ways. And like I told you in Chapter 2, what's really impressive is that the way that all of this comes together tells me who you are. Okay, I think that is really impressive, All of this is all the instructions to make everything about you, From your eye color to whether you're male or female, to whether you're going to get cancer or not. Pretty impressive stuff there. So we have those bases, A, T, C, and G, that pair up together. C always pairs with G, as you can see here, and A always pairs with T. So those always go together. And the sequence of these amino acids, are going to code for specific proteins. So let's take a look at what RNA is. So RNA is just one strand of the nucleotide, so just one of those halves of the DNA. And the bases here are a little bit different. We see our same A, C, and G, but here T is replaced by uracil, So instead of T, we have U. Basically what this is, remember, like I said, this is like your Xerox machine, This is your template. So what we're going to see here is A pairs with U, and C pairs with G. That C pairing with G is the same thing we've always seen, so we just replace the T with the U. But we have 3 types of RNA, 3 types of these templates, these copies. The first is that messenger RNA, that mRNA. And this one is going to carry genetic information from the DNA to the ribosomes. Okay, so again, this is like the messenger. It's carrying a Xeroxed copy to the ribosomes. Then we have ribosomal DNA. So this is in the ribosomes. What we have here is with protein, it's going to form new ribosomes and new proteins, So that's important. So that's definitely a function. So if we want to go back to function, messenger RNA, it's carrying the copy. So it carries the copy to the ribosomes. Ribosomal DNA, makes the proteins now. It has the code, now it's going to make it. And transfer RNA, tRNA, is going to carry those amino acids that we saw to the ribosomes where they join other amino acids to make proteins. So this is going to also help make proteins. Okay. So we need 2 processes, transcription and translation, that are going to help make those proteins. So the first is transcription. Transcription occurs in the nucleus, so that's important. It doesn't occur out in the cytoplasm, in the nucleus. So what we see here is that the DNA is going to code into the mRNA, okay, that messenger RNA. And then that messenger RNA is going to leave the nucleus and head towards the ribosome. So that's that first part, that we talked about here. Okay, that messenger RNA. So, as you can see here, we've got our DNA strands. So basically what we have, we have our DNA. It separates, and it's going to make a copy. So here, we start over here. We've got our 2 strands. The 2 strands are going to get pulled apart. So you see up here, this A would usually pair with this T, but remember in RNA we replace the T with the U, so the C would go with the G. These all pair, but this strand's going to kind of hang out, and it's making a physical, like, Xerox copy. Okay, so then what's going to happen is that copy is going to eventually break off, right, as you can see here, and it's going to head over to that ribosome. And once it hits the ribosome, it's going to sit in this protein here, and it's going to be read, essentially, okay, by a little reader. It's going to get run through there, and usually what we see is every 3 pairs makes for a protein. So what you can see here, you see these little amino acids. Each amino acid is going to be different, right? So you see you've got different colors here. Each one of these this 3 section of color is going to code for a different amino acid. So when it hits these 3, this amino acid comes down and picks it up. When it hits these 3, this amino acid comes down and picks it up. Okay, and here's some of your amino acids over here. So here that AUG is going to code for this amino acid. GGC goes for this one. So you can see it goes on, right? All these different amino acids. It recognizes that sequence, comes down, and picks it up. Pretty cool stuff. Pretty complicated. Then we have translation. So translation, right, is basically what we were talking about there, is where that messenger RNA is going to form that protein. So those 3 bases that are going to sit there, we call those a codon. So every 3, right, those 3 colors that we looked at, like A, U, and G here, that makes a codon. And basically then the ribosome is going to read it, and the anticodon is going to come down and pluck it out, and the amino acid is going to bind with it. So it's pretty cool, right? That's that second part, right, that we were talking about that happens. So pretty neat stuff.